Hello everyone, this presentation is on the introduction to lipids. So with regards to the structural role of lipids, we have two different types of lipids. We have the neutral lipids, and these are the lipids that have little to no charge, which means that they're not soluble in water. And an example of this includes glycerides and cholesterol esters. The other type of lipids are the amphipathic lipids, and this type of lipid has two parts. It has a hydrophobic water-soluble part and it has a hydrophobic water-insoluble part. And the amphipathic lipids are used in biological barriers, for example, the phospholipids in cell membranes. And before we get into talk more about lipids, and I wanted to take a moment to talk about the general structure of fatty acids. So on the left-hand side of the screen, you can see that this is the functional group of carboxylic acid. And basically a fatty acid is that carboxylic acid group attached to carbon atoms, which is sort of on that's our representation of the carboxylic acid is a chain of carbon groups right here. And that's why we call it a fatty acid. In terms of classes of lipids, we have two classes. We have the cholesterol and other sterols, and then we have the glycolipids. And we can see here on the left-hand side of the screen an example of a cholesterol molecule, which has a bunch of rings and other carbon atoms attached to it, along with a hydroxyl group. With glycolipids, glycerolipids, it is made up of a glycerol backbone, which is the pink part here, and it's attached to fatty acids. When we have three fatty acids attached to glycerol, we call it a triglyceride. But when we have two fatty acids attached to this glycerol backbone, we call it a diglycerol. And an example of that is a phospholipid, in which this third fatty acid is a phosphate. What was triglyceride would be a phosphate group. So whenever we have a phosphate group and two fatty acids, we refer to that as a phospholipid. And this is a molecular depiction of what a phospholipid would look like. And most of you would probably recognize that this sort of depiction of like the cell membrane phospholipids, where we have like a circular head and two tails. And right here in the middle is the glycerol backbone. And then we have the first two carbons of the glycerol backbone are attached to the hydrophobic tails. And then because of the phosphate group here, and it's a charged group, so it makes this head hydrophilic. In terms of these phospholipids, the fatty acids that are attached to this first and second carbon, this carbon right here is known as the SN1, and the second carbon is known as an SN2. And so the fatty acids here we can see are attached to the SN1 and SN2 position, and the phosphate group right here is attached to what is known as the SN3 position. In terms of classes of lipids, we just talked about the sterols and cholesterols, and these are made endogenously, and they're not considered. Cholesterol, for example, is not considered a source of energy. Our bodies use it as a structural component of the cell membrane to maintain membrane fluidity. It is also cholesterol is involved in bile production, so it's a very important part of the bile synthesis. It is also a precursor of many steroid hormones, for example, estrogen. Whereas the glycolipids, as we just saw from the figures, that when it's made up of a glycerol backbone attached to fatty acids, and when we have two fatty acids attached to the glycerol, it's called a diglyceride, and when we have three fatty acids attached to that glycerol backbone, it's called a triglyceride. And these are neutral lipids, which means that when we have especially the triglyceride, it's not doesn't have polar groups, so it's water insoluble. In terms of classes of fatty acids, we categorize them into 3 different categories based on length, which is the number of carbon atoms attached to that carboxylic acid functional group, into short-chain fatty acids, medium-chain fatty acids, long-chain fatty acids, and very long-chain fatty acids. The second categorization is based on saturation level. So this is basically the number of double bonds that are created by a class of enzymes known as desaturases. And the third classification is based on the type of double bond, whether it's cis or trans, and we'll get into more detail about each one of these. So in terms of the length of a fatty acid, for the short chain fatty acids, we have less, that string of carbon atoms would be less than 6 carbon atoms, basically 5. Anywhere between 1 and 5 would be our short chain fatty acids. The medium chain fatty acids have a string of carbons anywhere between 6 to 12 carbons long. And our short chain and medium chain fatty acids are both water soluble. And the very long chain fatty acids, it has 13 to 21 carbons. And the very long chain, the very last one, the very long chain fatty acids, are the ones that have 22 carbons or more. And the long chain fatty acids and very long chain fatty acids are water insoluble. And it is important to remember whether or not they're water soluble, because this becomes important later when you learn more about lipid synthesis and metabolism. In terms of saturation level, we have the saturated fatty acids and unsaturated fatty acids. And the saturated fatty acids, you can see an example on the left-hand side of the screen where that the string of carbons attached to the carboxylic group has no double bonds, and that's why it's called saturated fatty acids. The unsaturated fatty acids, we have 2 different categories. We have the monounsaturated fatty acids and then the polyunsaturated fatty acids. And the unsaturated fatty acids, as you can see from this figure, has, and as the name implies, has 1 double bond. Mono referring to that 1 double bond that you have, whereas the polyunsaturated fatty acids have multiple double bonds in that fatty acid chain. And here's just a more visual depiction of what we mean by saturated and the implications of, when a fatty acid is saturated versus unsaturated. And this is the a phospholipid, and we have this phospholipid head group, and then it's attached to 2 fatty acid chains, one of which is saturated, which you can see here has no double bonds, and it's a straight string basically of these carbon molecules, as opposed to the right-hand side string where you have a double bond here that introduces a kink. And you can see from this figure on the right-hand side, when you have saturated fatty acids, it creates this compact structure, and you can think of butter, like it makes it more rigid, basically, as opposed to when you have these little kinks, it makes it more fluid, and you can think in your mind about oils versus butter when you think about saturated versus unsaturated. So in terms of the double bond, the type of double bonds, we have the cis or trans. So on the very top of the screen, we have a saturated fatty acid that has obviously no double bonds. And then in the middle, and the bottom 2 molecules, we have unsaturated fatty acids, the middle one being a cis fatty acid. And the reason for why we call it a cis, this is sort of like a chemistry review, that the hydrogen atoms around the double bond are on the same side. If the hydrogen atoms around the double bond are on different sides, then we call it a trans fatty acid. And in terms of the nomenclature of fatty acids, we have 2 different types of naming systems. The first one is the common one, which is basically like a given name to a fatty acid. And this is, you just have to basically memorize the name of it. And then the second type is the delta and omega systems. And these are more methodical ways of naming fatty acids. And I'm just going to go ahead and we'll take a closer look at the delta and omega method of sort of naming fatty acids. With the delta system, it usually, when we come to write the name of the fatty acid, you just write a C that represents the number of carbon atoms in the molecule, and then it's followed by the number of the fatty acid lengths, which is basically the number of carbons in that fatty acid. And then you have a colon and then the number of double bonds from the acidic end. So in this figure, you can see here, this is our acidic end. So the first carbon is right here, numbered 1. And when you go down that chain, whenever you hit that double bond, that's where you write that double bond number after a delta. So you write delta with a superscript of where the double bond starts, and then you follow it by whether it's a cis or trans. The omega system is very similar to the delta, except that it starts, instead of starting from the carboxylic end, we start from the methyl end. So as you can see here in blue, that represents our omega naming. And here is an example of palmitic acid. And if we were to represent that using the delta system, it would be C16. And then you'd write that colon and number 1, which is the number of double bonds, which is right here. We have 1 double bond, followed by delta, and then the superscript of where that double bond is, which is at carbon number 9, followed by trans. And it's the same thing with the delta system, except that instead of writing a delta, you'd write an omega, and then you follow the omega by where that double bond is with reference to the methyl end as opposed to the carboxylic end, or carboxylic acid end, sorry. So this brings us to our next topic, which is essential fatty acids. The polyunsaturated fatty acids, or PUFA, which have a double bond at the omega-3 and omega-6, we refer to those as essential fatty acids. And the reason for that is that the human body cannot synthesize double bonds at the omega-3 and omega-6, so we have to intake that through our diet as opposed to our bodies producing it. Because the human desaturases, which are the enzymes involved in introducing double bonds, are delta specific, or they basically they work from the acidic end, and the human isoforms are the delta 9, 6, 5, and 4. The omega-6 fatty acids have high pro-inflammatory potential, and omega-3s have low pro-inflammatory potential. And in Western diets, we tend to consume more of the omega-6 fatty acids as opposed to omega-3s. And this is not entirely bad if you're going through an inflammation, if you've had like some sort of trauma and that caused inflammation in your body, but it's something to note for future reference. The omega-3 and omega-6 fatty acids, the most common types of omega-3 fatty acids are the ALA, EPA, and DHA. And of these 3 omega-3 fatty acids, the ALA are the most common of them, and you don't have to memorize you could if you wanted to memorize the actual names of these fatty acids. And then the common omega-6 fatty acids are the LA, or linoleic acid. So the brain has very high levels of omega-3 fatty acids. About 30% of the brain mass is made up of DHA, and this is believed to have a role in neural development. So this is it for my presentation. Here are my references for the figures and the information.